Hello, my name is Mark Larson. I'm a professor here at Augustana College. I am a cell biologist and biochemist by training, and specifically my research focuses on blood platelets. These are the small anuclear cells that traverse your blood vessels, and you have about 1 trillion of them in your body, and I'm really interested in what makes these things work. They serve as sort of cellular sentinels that respond to damage within the vessel to help repair and replace vessels that have been damaged. And part of that process helps normal blood flow as a matter of course, but it can also be a problem in cases like heart attack and stroke, where damage to vessels can actually result in the entire vessel being blocked off. So I study what platelets do, how they work, and what changes their function. And specifically, I've been looking at a couple different elements, and one of them is how omega-3 fatty acids, or fish oils that are found in things like salmon, how they influence platelet function. We've known for about 4 decades that omega-3 fatty acids are really good for you, they're good for your heart, and people who ate a lot of these fish tend not to die of heart attacks and strokes as much as other people do. And we're trying to get at in our lab as to whether or not these omega-3 fatty acids influence platelet function in a way that can help explain why people don't die of heart attacks and strokes. So my lab is particularly interested in how these fish oils alter normal platelet physiology. I'm also very interested in platelet biogenesis, the factors that go into how you replace the 1 trillion cells that circulate through your blood at any given moment. Those 1 trillion cells have a lifespan of about 7 to 10 days, so you're constantly making new platelets, and so I'm interested in looking at the factors that create platelets from their cellular progenitors that are found in the bone marrow called megakaryocytes, and the cues that they must get in order to make new platelets on demand. And so the work that goes on in my lab with undergraduate researchers involves these 2 projects. We look at the effects of different elements on platelet function, and we look and try and understand how platelets can be made and how they can repopulate existing cells within your body. And all the projects I do tend to be offshoots of those 2 general ideas, and the different specific projects we look at from year to year tend to vary, but they always focus specifically on platelets, platelet function, and how platelets get made.